This is the first lecture in Unit 7 for microbiology. There are not a whole lot of pictures that are in this unit, and all of the links are actually more related to things to read. So I went ahead and put the links as a separate page for you instead of integrating them into the document this time, since that seems to be more effective for your reference materials on this type of subject. Symbiosis refers to organisms living together. This relationship may be mutualistic when both organisms benefit. In commensalism, one organism benefits without significantly affecting the other. It is thought there are very few, if any, truly commensal relationships. With parasitism, one organism benefits while the other is harmed. A pathogen is a parasite that causes disease. The normal flora are organisms which normally colonize the body without causing disease. On the skin alone, we have about 3 million bacteria per cubic centimeter. The resident microbes are part of the flora throughout life. Transient microbes are on the body for hours to months before disappearing. As a fetus, we are sterile, but flora development begins as the infant passes through the birth canal. Additional flora is acquired throughout the first meal, additional meals, and skin contact. Opportunistic infections don't cause disease under normal circumstances, but may if the opportunity arises. A person who is immunosuppressed will be more prone to opportunistic infections. Immunosuppression may result from disease, malnutrition, severe stress, extremes of age, radiation and/or chemotherapy treatment for cancer, AIDS, or the use of immunosuppressive drugs for transplants. Altered flora may open the door for opportunists. Flora create microbial antagonism or competition. Changing this may allow some organisms to become opportunists. A common example is Candida albicans in the vagina. Introducing organisms into an abnormal location where they are not normally present may be problematic. Often these areas lack competition and growth occurs very rapidly. An example would be the introduction of organisms into the peritoneal cavity after surgery. In that case, there is no competition and the organisms are able to grow very rapidly and be potentially life-threatening. Contamination is the presence of microbes in or on the body. Infection is the result of successful invasion of a pathogen. Microbes need a portal of entry into the body. The skin provides a good barrier when it is intact. Some organisms are able to enter through natural openings like hair follicles or sweat glands, while others need cuts or scrapes to enter. Some organisms can actually burrow through the skin, and others can digest the outer layers to get in. The mucosa line body openings open to the outside world, such as the mouth and the conjunctiva. These surfaces are warm and moist, so they are more hospitable. Microbes may enter through the respiratory tract and even enter the respiratory tract via the eyes. They may enter the gastrointestinal tract in our mucous membranes. The placenta typically provides an effective barrier, with less than 2% of pregnancies having a pathogen cross to infect the embryo or fetus. Pathogens may be deposited parenterally beneath the skin or mucosa through cuts, bites, punctures, stabs, deep abrasions, or surgery. You may have heard of cases of infants being born to HIV-positive mothers and not being HIV positive at birth, and it just goes to show what an effective system we have to prevent fetuses from contracting diseases from the mother. It's not 100% effective, but it is a very good barrier that greatly helps to reduce disease passed from mother to infant. Microbes must be able to adhere to establish a colony. Adhesion is just the process microbes use to attach themselves. This involves adhesion factors, which are specialized structures or attachment proteins. The helminths use suckers and hooks, and some protozoa have adhesion discs. Viruses use ligands, which are lipoprotein and glycoproteins that enable binding to complementary receptors on the host. These structures are called adhesins in bacteria. An organism which is avirulent may have lost the ability to make adhesins or attachment proteins. When organisms form biofilms, they interact with the other microbes to form a web of bacteria and polysaccharide. Dental plaques are biofilms of up to 500 cell layers thick that are on your teeth. Disease is defined as injury significant enough to interfere with the normal functioning of the body, or a change from a state of health. Morbidity is the presence of disease, where mortality is the death rate observed as the ratio of deaths in the population. Signs are objective manifestations of the disease, observable or measurable by others. Symptoms are subjective characteristics of the disease felt by the patient alone. The objective nature of signs over symptoms can be really useful in a clinical scenario, especially if the patient's honesty about the disease is being called into question. Both come into consideration when making a diagnosis, although the signs are generally considered more reliable. A syndrome is a group of signs and symptoms that collectively characterize a disease or condition. In some syndromes, not all signs and symptoms are present in every case. Asymptomatic or subclinical cases may have no symptoms or are mild enough to go unnoticed. Etiology is the study of the cause of disease. The germ theory of disease states disease is caused by microorganisms. Koch's postulates are used to demonstrate a microbe is pathogenic and is the cause of disease. Koch's postulates state the pathogen is present in every case of the disease and may be isolated and grown in pure culture. The pathogen must cause disease when introduced into a healthy susceptible host, and the same pathogen re-isolated from the diseased experimental host. There are some exceptions and difficulties in applying the postulates in real-life situations. Some pathogens cannot be cultured in the lab, and in some cases only because they are too delicate. Some are caused by a combination of a pathogen and other sets of conditions where they may otherwise be harmless. This is the case with many opportunistic infections. It is unethical to apply all of the postulates, particularly to infect a healthy host just to confirm a pathogen. It is not always possible to establish a single cause for a certain disease, such as pneumonia and meningitis. Those diseases may be viral, bacterial, fungal, or parasitic in nature. Other pathogens were ignored for years, such as Helicobacter pylori. Virulence is the degree of pathogenicity, and pathogenicity is the ability of a microbe to cause disease. Pathogens have virulence factors or traits which enable them to enter the host, adhere to cells, and gain access to nutrients, and then escape detection or removal by the immune system. Extracellular enzymes are used to dissolve chemicals in the body and help maintain infection. Some examples are hyaluronidase to break down hyaluronic acid, collagenase to break down collagen, coagulases, and kinases. Microbes may produce toxins or chemicals to harm host tissue, or chemicals to harm and trigger the host immune response. The host immune response can cause damage in itself. Exotoxins can destroy host cells and interfere with host metabolism. Cytotoxins kill host cells or affect cell functions. Neurotoxins interfere with nerve functions, and enterotoxins affect the lining of the gastrointestinal tract. Antitoxins are antibodies that bond toxins and neutralize them. Endotoxin, or the lipid portion of lipopolysaccharide, the LPS in gram-negative cells, is also a toxin. Microbes can increase their virulence and combat host immune responses by resisting phagocytosis. One way is by forming a capsule. Many are composed of chemicals normally found in the body, so they do not stimulate the host immune responses. Capsules are slippery to make phagocytosis difficult and may slow digestion by the phagocyte. Use of antiphagocytic chemicals prevent the fusion of the lysosomes with phagocytic vesicles, allowing the bacteria to survive in the phagocyte. M protein is a protein on the cell wall and fimbriae of Streptococcus pyogenes that resist phagocytosis. Leukocidin is a chemical that destroys phagocytic white blood cells. The disease process often begins with an incubation period. This is the time between infection and the first signs or symptoms of disease. The length depends on virulence, infective dose, site of infection, patient health, and the nature of the pathogen. The prodrome is the short time of generalized mild symptoms that precede the illness. Not all diseases will have a prodrome, but it is the time when you feel like you are coming down with something. Illness is the most severe phase, when signs and symptoms are most evident and the immune system has not fully responded yet. During decline, the body gradually returns to normal, and immune responses and medical treatments attack the pathogen. In convalescence, the patient recovers from the illness. This includes tissue repair. The length depends on the amount of damage, the site, and the nature of the pathogen. The portal of exit is the way the organism leaves the infected patient in order to infect others. This is often similar to entry or through host secretions. Reservoirs of infection are sites where the pathogen is maintained as a source of infection. Animal reservoirs include both domestic and wild animals. A zoonosis is naturally spread from an animal host to a human, where vectors in animals are arthropods that transmit the disease from one host to another. Human reservoirs occur when the pathogen incubates in a human carrier. The reservoir may have an active disease or be asymptomatic. In general, a more successful microbe will not kill its reservoir, or at least will take a longer period of time to do so, allowing for more opportunities to spread. There are several modes of transmission, which can be summarized as the 4 F's: flies, feces, fingers, and food. Contact transmission is spread from one host to another, with direct contact involving body contact between hosts and a person-to-person contact spread without direct physical contact. Indirect contact sources include fomites, which are inanimate objects used to transfer pathogens to a new host. Doorknobs, faucets, and children's toys are all common fomites. Droplets may transmit disease while a person exhales, coughs, or sneezes. Vehicle transmission is carried through a substance such as air, water, food, or bodily fluids. Disease is considered airborne if it travels more than 1 meter in respiratory droplets via aerosols. Waterborne diseases use water as a reservoir vehicle and include many gastrointestinal diseases. Fecal-oral transmission involves organisms being shed in feces entering the host through the mucosa or the skin. Foodborne transmission involves pathogens present in contaminated food. Vector transmission is spread through animals or arthropods transmitting pathogens, often through bites. A biological vector transmits the pathogen and serves as a host for multiplication of the pathogen in some stage of the life cycle. Mechanical vectors are not required as hosts. They passively carry the pathogens on their feet or body parts. These include flies, roaches, and other similar organisms. So here we've got a picture of a fly walking on food. We don't know what the last thing was that fly walked on, but it could potentially pick up some contaminants that way. Epidemiology is the study of where and when diseases occur and the transmission within the population. The prevalence is the total number of cases, both new and existing, in a given area or population during a given time period. With diseases, we can classify them different ways. Acute diseases develop rapidly and last a short time. Chronic diseases develop slowly and may have continual or recurrent infections. The subacute disease has a duration and severity between the acute and chronic. In the latent stage, the pathogen remains inactive for a long period of time before becoming active. This also allows a greater opportunity for this organism to be spread from the host to others, increasing its population. Communicable diseases come from one infected host and are spread to another, either directly or indirectly. A contagious disease is communicable and easily transmitted between hosts. A non-communicable disease arises outside of the host or from normal microbiota. So when we look at the prevalence versus the incidence, prevalence is the total number of cases, both new and existing, in a given area or population during a given time period. The incidence is the number of new cases of a disease in a given area or population during a given time period. When a disease is endemic, it normally occurs continually at a relatively stable frequency within a given population, also within a given geographical area. A sporadic disease has only a few scattered cases within an area or population. An epidemic occurs when the disease is present at greater than usual frequency in that particular area or population. A disease is said to be pandemic when an epidemic occurs simultaneously on one or more continents. The CDC, or Centers for Disease Control, are located in Atlanta, Georgia, in the United States, and is the clearinghouse for epidemiological research. They publish the MMWR, or Morbidity and Mortality Weekly Report, with the statistics of all nationally notifiable diseases. An example table of nationally notifiable diseases can be found in this section of your notes here. And so here we go. That one's coming up. So this is as of 2005, but you could certainly pull up one that is a lot more recent. It allows you to see what month that it's caused in, so you can see if there's seasonal changes with that as well. And occasionally the list of diseases that is on this list will change a little bit. Epidemiological studies observe the dynamics of a disease in the population. Descriptive studies tabulate data concerning a disease of key importance, which is the index case or the first case of disease in a given population or area. Analytical studies investigate the disease details to determine probable cause and the mode of transmission. Prevention is often studied retrospectively, but is useful for future incidences. Experimental studies are geared towards testing a hypothesis concerning a disease, such as testing treatments or prevention techniques. Nosocomial infections are those acquired in a healthcare facility. An exogenous nosocomial is caused by pathogens acquired from the environment. Endogenous nosocomials arise from normal microbes within the patient, but due to factors within the healthcare setting. These are often opportunists. Iatrogenic infections are the direct result of medical procedures, such as catheters, surgery, or invasive diagnostic procedures. The microbes allow others to thrive and can result in superinfection. Superinfection and antimicrobial resistance are very serious concerns in healthcare facilities. The occurrence of nosocomials is influenced by several factors. For example, in a healthcare setting, a person is exposed to numerous pathogens, and many are antibiotic resistant. Many ill patients have weakened immune systems. There is transmission of pathogens among staff, patients, and visitors via routine activities of the staff. This includes invasive procedures, use of sterile instruments, and the use of protective barriers against blood and body fluids. There are branches of public health organizations from worldwide down to local communities. Local public health is primarily run by the city and county health departments, who report their data to state agencies. At the state level, reporting is governed by state law. States will play a role in epidemiological studies and publish a bulletin similar to the MMWR. The CDC is one national branch. They conduct research in etiology and prevention. The WHO, or World Health Organization, coordinates efforts to improve public health throughout the world. Standards of cleanliness are essential to interrupt disease transmission. Potable water is necessary to have water fit to drink. This is achieved through filtration and chlorination processes in most communities. Standards are set in the food industry for canning, preserving, pasteurizing, and irradiating food. Techniques are established for food handlers. Vector control, especially for mosquitoes and rodents, is necessary. This often involves eliminating breeding grounds and use of insecticides. Personal precautions such as washing hands before and during food preparation, disinfecting kitchen surfaces, proper refrigeration and freezing, and adequate cooking of meat are very effective. Also, public health education to help the public make healthy choices. Some things are more difficult to control, like sexual and air transmission of diseases.